She might be out of her.
Are we ready, or is there anything?
No, not ready.
Oh.
I say we're ready. You say
If you want to start it.
Might as
We're
well
starting
get
on
going.
time. We're going to
Yes,
stay on
let's
time. Good morning, everyone. First, today we have Susan Elias with Deer Tick Phenology and Warming Climate in Maine.
Okay, so I want to talk about phenology in the context of climate change. And phenology is, when do the lilacs bloom? We're seeing that earlier now. When do the goldfinches arrive? Is it dark enough? It seems a little bright in the front here. Yeah. So many of you have seen this before. This series of maps depicts the range expansion of the deer tick in the state of Maine over the past several decades. And so the deer tick is an invasive species. It has invaded from the south, and the warmer colors depict increasing abundance of the deer tick. So range expansion plus increasing abundance. These data come from the Maine Medical Center's Vector-Borne Disease Lab, and these are ticks off of people and pets. And this is a data set that I'm going to be working with and showing you this morning. So I took this data set and I basically divided it into the northern tier and the southern tier, being the northern 5 counties and then the rest of the counties to the south. And we know that that's not a real even north-south division, but this is how I started out back in 2014 looking at some of these data. And what I did here was organize it by the early decade and then the later decade. So again, we stopped taking data after 2013. And so then I also divided it north and south, as I said, and I asked the question, I said, has there been any change in phenology? Do we see a difference in peak week of nymphal activity? So we're looking at nymphal deer ticks here. And the problem with nymphs is what?
They're little.
They're tiny, you can't see them. So when do most human cases of Lyme disease arise? Like spring, summer, or fall?
Fall.
Actually summer, because the nymphs come out in the summer. They peak in the summertime. And like you said, Cindy, they're tiny, so we don't see them, so we get Lyme disease. But what you see here is a shift from week 26, which is about mid-July. This is in the southern tier, okay, to week 24. So that's a really big shift from the middle of July to late June / early July. So wanted to look at this more closely, and the question is kind of the research question for right now is to what extent is the deer tick invasion due to climate change? And we can expect linear relationships, and we can probably expect some threshold relationships. And I like asking and answering the questions about nymphs and disease and stuff because it's an opportunity for outreach, right? But I would not miss an opportunity to show you disgusting pictures. Okay, so up here we've got a gravid female, a blood-fed female deer tick. And the thing is, she's not going to start laying these 2,000 eggs until it's over 6 degrees Celsius. So if you put a gravid female in the fridge, it's about 4 degrees Celsius, she's not going to lay eggs. So once you hit that threshold, things start happening. And in the southern tier of the state, you get this egg laying in about May. Now you got to add degree days over 6. You got to accumulate those degree days. And once you do that, and once you hit about 1,200 degree days, you get larvae. Can you see the little larvae hatching out of those 2,000 eggs? So that's an important threshold. That's based on my boss Pete Rand's tick pot study. He put plaster buckets at locations around the state, and he put gravid females into those plaster buckets, and he counted up the number of days it accumulated to that larval hatch. We think that's an important threshold. Now, these larvae have got to come out towards the end of August because they're racing the clock. They've got to find an animal to blood feed. And this is like a small mammal. This is a white-footed mouse ear right here. And you see little larvae feeding. And then what's got to happen before wintertime hits, this is not a mating tick. This is a nymph molting from the larval stage. That's got to happen before wintertime because you get low survival and even death when temperatures get below. This is the this is like a lower threshold for survival and thriving. Minus 14 degrees Celsius gets to be hard on these ticks, even the nymphs, which are very cold hardy, right? So, here we go. So Sean did these beautiful degree day accumulation maps. And in the warm colors, what this signifies is that you have reached that 1200 degree threshold by the end of August. But if it's a cool color, if it's the green, then you're not going to get there's not enough heat being accumulated to get that larval hatch. All right. But if you look at the current decade, the decade that we're in, you can see how that warm zone has moved up. So another threshold, the other one I touched on was that low temperature of minus 14 Celsius. And if we just look at a difference map, you know, this decade versus, this is a 2-decade span, and we just go to our awesome climate reanalyzer and we take a look at the warming winters, we can see that, yeah, the whole state is warming in general, but we've got even more warming in the north. So this is sort of that northern versus southern tier that I'm talking about, trying to compare them. So again, you know, we've got this situation of warmer winters, and let's take a look at the linear and threshold relationships between that and deer tick abundance. All right, so let's go. My dad said, don't drag them through this, but I'm going to drag you through it. So we've got the northern tier and we've got the southern tier. So in blue, we've got, this is nymphal abundance, okay? And then in the red, we've got this minimum winter temperature, December, January, February. And I've also got this black line marking that minus 14C threshold, and you can see that we're below that in the earlier decade, but we're starting to poke above it. And so if you look at the southern tier in contrast, you know, many, many more nymphs in blue, and you can see that the minimum winter temperatures are above this minus 14C threshold. Is there a linear relationship? The trouble with an invasive species is in the beginning of the invasion, there's lots of suitable but unoccupied habitat. The other thing to point out here is in 2000, year 2000, there's this amazing spike in nymphs. These nymphs came into our lab, we said, what's going on? Now there's a series of warmer winters here, but could that explain that spike in nymphs? Well, there's more than one driver of deer tick abundance, and I'm going to talk about deer. And another threshold, there's two camps in my field of vector ecology, tick ecology. And there's a group of us who feel, based on empirical observation, that there's an important threshold above which, if you get to deer densities of over about 12, maybe 8, 13, 15 per square mile, something like that, you're going to get more ticks and more disease. So if we now superimpose that nymphal time series over the deer, here's that threshold of 15 per square mile. I mean, you could move it around, but you're only around 5 per square mile in the northern tier of the state. And this little dip right here represents a harsh winter. So according to inland fisheries and wildlife biologists, this is why the deer herd actually dropped. Okay, but if you go over here, look at the deer herd here. Look at that 15 per square mile threshold. And look at that spike. This is the mild winter, according to biologists, deer biologists, that allowed the herd to spike up to about 23 per square mile. But actually, you know, I didn't add error bars there, but in one wildlife management district, it went up to almost 50 per square mile. Now look at this. That if these deer are feeding adults and subadult ticks, that could lead up to that spike. So putting it together in a model is tricky. But what I did is I used an autoregressive model where I was allowing nymphs to be a function of themselves and temperature and deer to up to 2 past lags. And what I found in these models was that there were in the northern tier, more nymphs were associated with warmer winters. Didn't do so well with the degree day accumulation, but not deer in the north. But in the south, it was all about the deer. And also these models don't really explain a huge amount of variation, maybe about an R square, pseudo R square of about 0.3. So more work to be done. That's it for now. you have any questions at this early time?
Next, we have Annie St. Amand with Climate-Driven Migration, Prioritizing Cultural Resources Threatened by Secondary Impacts of Climate Change.
Thanks, Will. All right, I see the 1 minute left sign is already up, so I'll be brief.
We want all the minute details.
Yeah.
The minute details.
All right, hi everyone. My name is Annie, like Will said. And today I'm going to be discussing some research that takes place, like Susan's here, in Maine. While during my dissertation, I look at abrupt climate change and human adaptation in Peru. Today I'm going to be talking about climate-driven migration here, which is an impact of climate change. So everyone in this room is well aware of the primary impacts of climate change. We have sea level rise and coastal erosion, as well as peat slumping, freeze-thaw cycles, storm surge. Here, let me point out, we have our very own Alice Kelly. She and Jackie have recently defended a very successful Lost to the Sea project for Jackie's dissertation. Congratulations. Today, I'm going to be talking about what I define as a secondary impact of climate change, which is climate-driven migration. Now, the United Nations predicts increased resettlement of climate-affected communities over the next century, and that's the time scale for this project that I'm working on. This is resettlement from areas where adaptation is not economically feasible, legal, or physically possible. So here we are. Southern Maine is where I've chosen for the pilot study for this project. Archaeological sites. So there are archaeologists in this room, and if you're not an archaeologist in the Institute, it's likely that you've at least collaborated with one. So many of you know that archaeological sites represent past human activity in the landscape. Hello. These are settlements, hunting sites. I can do it without the mic also, I'll just speak up. Artifacts, human and faunal remains, earthworks, and modified landscapes. But archaeological sites also represent important paleo-environmental and climatic proxies, which we as climate scientists can use to understand the past. They also represent dynamic landscape change. So the objective of this research, I wanted to develop a method to identify and prioritize highest risk sites, archaeological sites. And the framework I used was a quantitative risk assessment matrix. The significance of this method for identifying risk to cultural resources like archaeological sites is that it's a rapid method and that it uses freely accessible data so that communities and state and federal officials can easily identify risk to archaeological sites and mitigate this risk prior to population increase or the increase of infrastructure associated with resettlement. So some of the data I used for this project, and this was a little complicated because I had to draw from many fields to develop this framework, were archaeological surveys and predictive site models. The data were also economic. I looked at things such as municipal tax records, socio-demographic, such as US Census records, geographic. I put a lot of this together in a GIS system, or Geographic Information System, as well as land use and cultural resource management regulations at the town level, at the state level, guided by the Maine Historic Preservation Commission, as well as laws about construction or reconstruction in wetlands or shoreline zones, as well as at the federal level. Think about things like the National Historic Preservation Act, or the National Flood Insurance Act, which I'm sure you all think about often. So here's an example of a quantitative risk assessment matrix I developed. This risk matrix, like I said, identifies towns that are more likely to experience increases in climate-driven resettlement from present to 2100, thus posing a risk to archaeological sites. Along the top, you see factors which make a town more appealing for relocation. Like I said, this is just a sample. My pilot study area has 53 towns. Along the left, you see Durham and Cape Elizabeth. So for something like low property taxes, which is an economic incentive for relocation, we see that in this equally weighted matrix, 1 signifies a risk, whereas 0 signifies no risk. Other factors were more complex, like the last two you see to the right. I had to combine things such as predictive site modeling, parcelization and land development trends within the state, as well as cultural resource regulations. And then I included these figures, and if nothing else, maybe you'll appreciate the fact that they're not tick pictures. Sorry, Susan. Really enjoyed your talk. But here we have from the US Census trends in population change versus trends in housing unit change. These are divergent trends, just highlighting the importance of looking at multiple factors when trying to understand resettlement and migration. This allows, like I said, for the examination of cultural resource vulnerability. So the product of this research are maps at the town parcel level, as well as at the county level. Today I'm showing you results at the town level that identify and prioritize cultural resources, as I said, threatened by secondary climate impacts. And one of the key parts about this research is that it's applicable to similar coastal regions throughout North America. I wanted to develop a framework that will help communities assess risks to these important resources. And like I've touched on, the audience, researchers, communities, government officials. So here's an example of a scaled risk map produced using this method. It identifies towns where archaeological sites are most threatened. And along the right, you can see the scale of risk, low to high. And again, as I said, this allows for prioritization by town and by site. This is important over the next century, as coastal areas like Casco Bay are going to be experiencing impacts of climatic change more rapidly. So next steps for this project, I'd like to apply this model to other coastal regions using publicly available data. The Digital Index of North American Archaeology, developed over the recent years, is an excellent resource for archaeological site data. And like I said, the other data is freely available. And I'd like to use this tool to communicate risk to town and state officials. I've been working with the Maine Historic Preservation Commission, Arthur Spies, the state archaeologist. And another thing that I'd just like to touch on briefly is that I strongly believe that public participation in this process equals stewardship. It's really important for us to incorporate the public into work like this and to communicate the results of science to the public in order for our research to be effective. I'd like to thank everyone for listening, and I'll take any questions if there are any. Susan, yes.
I was just wondering about the tax burden assessment. So like Cape Elizabeth was a 0 and Durham was a 1. So how are you looking at that tax burden?
So for that, understanding it as an incentive for relocation, I developed, I calculated a average property tax income threshold. And so areas like Cape Elizabeth with higher than average property taxes were not deemed to be at risk for this one factor for relocation, whereas Durham with lower than average property taxes this is likely to be an incentive. Just one of the factors that I looked at for relocation. Thank you.
Next, we have Kisei Tanaka. He's going to present on a model-based approach to incorporate environmental variability into assessment of an American lobster fishery.
Morning. My name is Kisei. I am a fishery scientist and fishery ecologist. My research mainly focuses on counting the number of fish in the ocean. And when I tell that to my parents, they're like, what? Anyway, that's my research. As you can see, mostly I focus on American lobster. That's very popular in these areas in Gulf of Maine. But today I would like to talk about this model-based approach to incorporate the climate change, environmental variability into assessment management of lobster fisheries. So just to give you a quick background, this is the how the lobster fishery in the United States has developed over the last 50 years. You can see the trend has been increasing. This is landing. And compared to this point 1915, 2017, this is about 300% increase in landings. Right now, the lobster fishery is the most economically valuable fishery in the United States. The second most valuable fishery is scallops. $669 million US in 2016, about 97% came from Gulf of Maine. So, and as many of you know, that Gulf of Maine currently experiences very abrupt change in ecosystem due to climate change. So this is the just the one figure shows the sea surface temperature trend 1982 2016. This color indicates the warming trend compared to the rest of world oceans. Gulf of Maine here currently sits at 97th percentile. So if you just compare this part to the rest of oceans, they're very warming much faster than the rest of oceans. So this is from 2017 figure Gulf of Maine Research Institute scientist Andrew Pershing. The problem is that the lobster management currently only focuses on the harvesting rate and the spawning stock biomass, but do not explicitly incorporate this change in environment, which we already know that already affecting lobster populations. So this is where my research comes in. I mostly use mathematical models to count the number of lobsters in oceans. So what I use is what we call the population size structure population models. Why we call the size structures? Most of the time, fishery scientists try to group the fish by age, but we can't age lobsters because they molt. So what we use is this size-based approach to count number of lobsters in your stock unit areas. So this is number of lobsters in next time step is a function of growth, survivorship, current lobster populations, plus number of lobster coming in to the fisheries at each time steps. So most of the time I'm very interested in this, is this number of lobster coming in fisheries, recruitment, is affected by change in environment. So that's the part that I'm trying to modify this equation by trying to incorporating environmental variability index. So this is just my study areas. So this is the about the University of Maine. This is the Gulf of Maine. It's by NOAA definition, the Gulf of Maine lobster stock unit is about this part, and this part is what we call the Georges Bank. Used to be separate, managed by separate unit, but now it's combined unit. And this is the type of data that I use, being collected over 30 years. So this is the bottom troll survey conducted in this area between 1984 2013. So most of the time, I try to use this data collected by state and federal agencies and uses these mathematical models to estimate the parameters as a fish mortality, number of lobsters, harvesting rate, and the number of recruitment coming in each year. So by doing this parameter estimation, I get the output. The model output tells you what are the status of fisheries, what are the population estimates, how many lobster dying, how many lobster joining, how many lobsters growing into the next size classes. Interestingly, that I find out is that when I try to use the bottom temperature, bottom salinity, the climatic variables, it was harder to incorporate into the single index. So I used what we call the habitat suitability model that I developed a few years ago. So this is a species distribution model that can quantify the quality of lobster habitat as a function of temperature and salinities. You can see these spatial variations in lobster habitat suitabilities. So this is a suitable habitat, not so suitable habitat in the deeper waters. We can also quantify change in habitat suitability as a function of change in temperature and salinities over 30 years. So this is the how the lobster habitat quality have changed over years. Red shows the improvement. The blue is a decrease in habitat qualities. So this is the kind of time series lobster habitat suitability. And I use both the non-smooth time series or 3-year average or 5-year average. When I compare this time series to the recruitment deviations, they were highly correlated, positively correlated at zero lag, meaning that if suitability changes, then most likely that the recruitment is affected at the same time steps. So I tuned lobster recruitment deviation using this habitat suitability time series. Result is that we see a slight change in the default model output. So this is a recruitment, number of lobster reaching harvestable size in each year. And you can see the base model that did not incorporate the environmental variability. Compared to the base model, the environmental model slightly estimate higher number of lobsters coming into the fisheries in these recent years. At the same time, because the more lobsters coming in the recent fisheries last 5 years, fishing mortality was also lower, estimated by environmental models. And this is very important because most of harvesting guidelines in fisheries are based on these two parameters, lobsters. Then another thing is the what we call the retrospective analysis, that when you run the model with the different quantity of data to see if model can still produce consistent estimates. So you can see this, the number of peels, and if the model gives you a perfectly consistent estimate, you don't see any deviations in this model output. So this is what we call the Mons rho. 0 is good, larger this number, not good. So we compare this, the base model, the environmental models, you can see the Mons rho decrease in each cases. So this is the recruitment, Mons rho decrease from -0.11 to -0.09. Fish mortality also estimate 0.16, decrease to 0.13, meaning that by incorporating environmental variability in the model was behaving more consistently compared to the base models. So the implication is that population models can estimate the present state and past fluctuation key fishery matrix, such as population estimates. We can also use this model to account for the environmental influences on population processes. And these output currently being presented to the Lobster Technical Committee in Woods Hole can be used to improve the precision and robustness of stock assessment, changing environment. And I'd like to thank my funding agencies and colleagues. I'll take any questions.
So were you implementing these models in R?
Yes.
What were your main packages?
I have about 74 packages, but most of the time the species distribution model package is the SPE, and I have my own package that I wrote myself. And also I'm happy to share all these code and packages that you're interested in doing it. And I use a little bit of MGCB packages too. And population model is written in C++, but you can run in R. And and because the reason is that R is a bit slow in chunking all those numbers. The fishery data tends to be huge, but C++ is much faster in digesting these information, but R is much better in visualizing output. So
I'm wondering what the interpretation is of the divergence in different fronts that only occurs near the end of the time series.
right, so I can just show backup slides here. No, I don't. Never mind. So oh, there you go. So this is the base model output, recruitment, the model estimate, the change in number of lobster coming into the fisheries. The model was having trouble explaining this burst in lobster coming into the fishery in last 10 or 8 years. And model was able to explain this historic trend by adjusting its estimate with this part, the increase in habitat suitability. So by tuning this lobster recruitment deviation by the time series of environmental index, the model was able to slightly adjust its estimate at the end of time series. So that is what I guessed, but I am still trying to find out the other potential reasons. But in the model calculation, that's what's happening as of now. Thank you.
Next, we have Kit Hamley presenting on using paleoecology and archaeology to rewrite the human history of the Falkland Islands.
Awesome, thanks for the introduction. So I am a PhD student here at the Climate Change Institute working in Jacqueline Gill's lab. And a lot of what I'm going to be talking today is based on some of the work I did here for my master's project. But there is a bit of new information, and most of that's coming from a recent trip that I took to the Falklands, where we did some archaeological work. And my hope is that I can convince you with the evidence that we found the first archaeological site or prehistoric site in the islands. So for a bit of review, the Falklands lie some 300 miles off the southeastern coast of Argentina, in the cold, rough waters of the South Atlantic, and within the path of the Falklands Current, which is a branch of the circumpolar current that flows around the tip of South America and directly at and around the Falklands. And this is a nutrient-rich current that would have made it pretty easy for people to get back and forth from the mainland to the Falklands. The Falklands are also a biodiversity hotspot. They're home to some of the world's largest penguin, fur seal, seabird, and sea lion colonies. So it makes for really fascinating and fun field work. So when Europeans first arrived in the Falklands in the late 1600s, there were no human inhabitants. So it's basically always been assumed that Europeans must have been the first people to colonize the islands. At the time of European arrival, there was a single terrestrial mammal, a fox, locally known as the wara. So there weren't rats, there weren't cats, there weren't mice, no llamas, all of those things came later. It was just this fox. And the presence of a terrestrial predator on an island dominated by ground nesting seabirds, where there aren't other terrestrial animals, has led to some speculation that perhaps Europeans weren't the first people to colonize the islands, and that it might have been introduced by pre-European individuals coming over from South America. And yet, prior to this study, there have been no investigations into the prehistoric human history of the islands. And part of this might be because no archaeological sites have yet been identified in the Falklands. But that doesn't mean that humans weren't there. Establishing a reasonably firm date for the earliest human arrival on oceanic islands is often difficult because colonizers tend to be very few in number, which makes their presence very difficult to detect archaeologically. So in lieu of archaeological evidence, what we can do is we can turn to the paleoecological record and look for indirect indicators of initial human arrival. Even small numbers of individuals can have large environmental impacts, especially in island environments, which tend to be a bit more sensitive. And these impacts tend to come in the form of species introductions, perhaps like the wara, eradication of local species, and changes in fire regime. In particular, several studies have shown that charcoal, which can be used as a proxy for fire frequency and intensity, tends to greatly increase when humans first arrive in locations. Basically, we get to a place and we start setting things on fire. So for my master's project, what I did is I collected peat cores from throughout the Falklands and reconstructed fire history. So this figure shows cumulative fires from 3 locations in the Falklands. Mount Usborne is in blue. It is the highest elevation location in the Falklands and would have never really been utilized or by humans. So it acts as a bit of a control for recording regional signals, as it wouldn't have been affected by human modifications. We have Bleaker Island in red, and it shows a very similar fire history to Mount Usborne. And then we have New Island, which is in black, and you can see that it has a very unique and different fire signal than what we're seeing at Bleaker Island and Mount Usborne. Basically, there are very few fire events, and then all of a sudden, at about 1,000 years ago, fire frequency suddenly increases. So here we're looking at the fire record just for New Island. I'm sorry if this figure is a little difficult to see. So we have calibrated years on the x-axis, getting younger as you move to the right, and char, which is a measure of fire intensity, on the y-axis, which is also shown by these gray bars. Individual fire events are marked by pluses at the top of the figure. So if we take a look at this record, what we see is that for the first 7,000 years, there really aren't any fire events, or there are a couple, but they're very small and difficult to detect. So if we zoom in on the past 1,000 years, however, the fire record gets a bit more interesting. As you can see, there's an increase in both fire intensity, i.e. the gray bars are getting bigger, and fire frequency, so that the pluses are getting more frequent. And that starts at about 1,000 years ago. In particular, at 550 years ago, char magnitude dramatically increases. It's orders of magnitude greater than anything that we've seen on New Island up to that point. There's a brief hiatus in activity and then another increase in magnitude at roughly 200 years ago, which is when Europeans first started heavily utilizing New Island for whaling and sealing operations. So the big question is, what's causing this first initial major increase at 550 years ago? And since we're only seeing this change at New Island, what we can fairly confidently rule out any regional scale variation in climate or environmental factors. You would expect to see a signal similar to this at Mount Usborne and Bleaker, but we're only seeing it at New Island. So it also doesn't seem to be being driven by changes on a more local scale. And in terms of fuel load, which we can look at through loss on ignition, there's really nothing to indicate that that's having an effect on this fire. So instead, this 550-year event seems to have a very similar signal to the historic European occupation of New Island, leading us to believe that this 550-year event and perhaps events, or perhaps even the earlier events starting at 1,000 years ago, may represent initial pre-European colonization of the islands. So in 2015, we discovered 7 bone heaps on the north end of New Island. We dug a 1 meter by 1 meter test pit and collected bones for radiocarbon dating. We had to establish a marine reservoir correction age for marine mammals for this region of the world. And once we did that and applied it to the bone dates, we determined that the pile we sampled was 550 years old. So if you remember, this is the exact timing of that massive fire event that we're seeing on New Island. So of course, we had to return this past February to map and fully excavate the bone piles to determine whether or not they're archaeological. All of the bone piles are within walking distance of fresh water, to food sources, i.e. penguin and seal colonies, to driftwood beaches. There aren't any trees in the Falklands, so that would be a key component for fires, to shelter from the wind, and to gently sloping beaches. So we're able to fully excavate pile 6 and pile 7, which you can see highlighted there. Each pile was roughly 2 meters by 2 meters, and contained similar species assemblages to one another, which were primarily composed of rockhopper penguins and southern sea lions. Rockhopper penguins are a prey species for southern sea lions, so this assemblage is important as these two species are unlikely to naturally die in the same location on top of each other. The piles were located upslope from a sandy beach, and on average were about 12 meters above sea level. The bones were primarily located, as you can see, in a sandy matrix and were quite well preserved. So the excavated bones were carefully examined for cut marks, and specific bones were kept aside for determining the minimum number of individuals in each pile. Bone pile 6 contained 68 sea lions and 13 rockhoppers, and bone pile 7 contained 67 sea lions and 91 rockhoppers. These enormous numbers of individuals in each pile again lead us to believe that it's unlikely that these piles would have occurred naturally. We didn't observe any cut marks or cultural modifications on the bones, and we didn't find any associated tools. However, we did find a stone point was found in close proximity to and downslope of bone pile 7 in 1979 by a landowner and a local naturalist. The stone point was found on the surface and not in direct association with the bone pile, but is made of locally sourced Falklands quartzite and is consistent with lithic technology that was being utilized in Tierra del Fuego at this time. So Southern South America has a rich history of fox domestication, which I'm not going to go into in great detail because I don't have time. But in order to determine the origins of the war, I had to figure out how long the fox had been in the islands. So I sent off some bones for radiocarbon dating, and it turns out the fox had gotten to the islands by about 4,100 years ago. So while this arrival date predates our best information about human arrival to the Falklands, it doesn't rule out human transport. Rather, it could perhaps strengthen the case that humans had arrived prior to what we're seeing on New Island, and we just need to look for older sites. All right, so in summary, there are a number of factors leading us to believe that this is the first prehistoric site in the Falklands. First, the presence of 7 discrete bone piles composed primarily of species that would not choose to die together. Second, the close proximity of the bone piles to the location where Stone Point was found decades earlier. Third, the near synchronous timing of the orders of magnitude increase of fire frequency and the formation of the bone piles. And lastly, the presence of a single terrestrial predatory mammal species on an isolated island system where there are ground nesting seabirds. Together, these factors suggest that there was a prehistoric human presence in the Falklands, which pushes back the timing of human arrival to the islands by several centuries and rewrites the human history of the islands and what we know about exploration of the South Atlantic. So with that, thank you to everybody who's been involved. Thanks for listening, and I'd be happy to take any questions. Yeah.
So it's really intriguing, but it's very concerning that there's no modification and no tools associated with these bones. So my question is, are there any ethnographic cases of processing sea mammals without hard tools and without affecting the bones at all?
That's a good question. And
I think you're going to have to find that to make the case.
Yeah, I agree. It's concerning. I haven't come across any where there aren't associated tools within the middens or the piles. One thing that I think potentially slightly helps the case is that initial colonizers on islands are perhaps going to be a little more careful with their tools than people who are established. That being said, we still need to find tools that are in direct association, and we only were able to fully excavate 2 of the piles, which means that there are 5 more piles that need excavation. So the hope is to try to get back in the following year and do a bit of follow-up work and go from there.
But I would look more into the ethnographic record
Okay.
of sea mammal hunters and see if there's any way they could have been using these that's ethnographically known that would produce a record like this.
Okay. Yeah. Great.
Strengthen the
Thanks.
case.
Yeah, Annie?
I was wondering about the lithic tool. Is it sourced from local material, or is it similar to any assemblages off the coast of Argentina?
It's sourced from local Falklands quartzite, which is pretty neat. There was also another stone tool that was found basically a couple islands away that was the same lithic technology, but made from material sourced from a place in the Beagle Channel. So that's pretty neat. But yeah, that actual stone tool was made of locally sourced quartzite. Yeah.
Yeah, it's great to see how far you've come with this research over the last few years.
Thank you.
From the first war discussion.
Thank you.
Now, it's also intriguing that the location you found not only is a logical location, but one of the closest points to the South America.
Yeah.
Are you aware of any other areas where there are what appear to be bone heaps?
Yeah, so that's a great question, and one that I need to follow up on a bit more. I've talked with dozens of locals who have all said that they've never seen anything like these piles anywhere else. We fully walked like all of New Island and didn't find any other piles. The one possibility for a way that this can naturally occur is sometimes the peat will burn deep into the ground when there are fire events. And there are reports that animals can actually fall into these pits and accumulate. And at this point, that's all hearsay. I haven't seen them, but that's one of the plans that I have, is to go and try to find those naturally occurring piles and see if the number of individuals in these holes are comparable to what we're seeing here, and see if there's kind of any other places where these piles are showing up. But at this point, this is the only location. So thanks. Yeah, sure. Yeah, thank you.
What are the natural sources of ignition for fires?
It's lightning is the only one at this point. And the amount of lightning strikes are pretty low. Although there's not great data there. I wish there was a little bit more information about about that. I'm being told to clamor, but I'll catch up with you later and talk about that more. Thanks for the questions.
Next, we have Vanessa Hensley presenting on squirrels on the move, the response of the North American flying squirrels to rising ambient temperatures.
All right, so hi everyone. Thank you for joining me this early morning as I talk a little bit about my master's research involving flying squirrels here in the state of Maine. North America is home to only 3 species of flying squirrel, the third of which was very recently discovered within about the last year or two in the Pacific Northwest. Because this species is so new and has a pretty limited range, I've chosen to focus on the 2 more prevalent species, Glaucomys sabrinus and Glaucomys volans, or the northern and southern flying squirrel, respectively. As you can see from the range maps that I have up here, they are pretty accurately named. And Maine, which is denoted by the small star up there, is uniquely situated within the range for both species, making it an excellent study site for flying squirrels. What these maps cannot show, however, because they are just static images, is the northward range shifts both species have undergone over the past few decades. Range shifts we have even observed here in Maine. So here's a graph taken from a paper published by Connor Wood, who previously completed his master's here at the University of Maine. He analyzed long-term capture data from Holt Research Forest down in Aroostook, Maine, which is about 2 hours south of here. The blue line on here shows the abundance of northern flying squirrels, and the red line the abundance of southern flying squirrels. So prior to 1986, Holt Forest only had northern flying squirrels, but over a period of less than 20 years, a complete turnover from the northern species to the southern species took place. Connor suggested that climate change mediated by local forest turnover might be behind this dramatic shift. So this got us thinking, could climate change in fact be a direct cause of the observed range shifts that we're seeing? And are we seeing similar turnovers further north? With those questions in mind, we decided to focus on one specific aspect of climate change, rising ambient temperatures. The image I have up there now is probably quite familiar to most of you. It comes from CCI's own Maine Climate Future 2015 update and portrays the mean monthly temperature average across Maine for 3 time points, past, present, and future. It's no surprise to anyone here that we've already experienced temperature increases here in Maine, and predictions indicate even higher temperatures and longer warm seasons. So we hypothesized that rising ambient temperatures may be too energetically costly for northern flying squirrels, forcing them to move northward to cooler climates, the southern flying squirrels, already well acclimated to warmer climates can then expand their range northward to newly unoccupied territory. To investigate this hypothesis, the first thing we needed to do was determine how much it costs in terms of energy to live in a hotter Maine climate. We do this by measuring metabolic rate and evaporative water loss of resting flying squirrels as they experience different ambient temperatures. Doing so allows us to create graphs like this. We have ambient temperature across the x-axis of both of these graphs and a measurement of metabolic rate across the y-axis. And metabolic rate can pretty much just be thought of as energy usage. So when exposed to very cold or very warm temperatures, a mammal must use increasing amounts of energy to maintain the desired body temperature. So either heat itself up or cool itself down. Between these two extremes kind of exists a sweet spot known as the thermal neutral zone. Within this zone of temperatures, a mammal does not need to use extra energy to maintain its body temperature. So you can imagine it's therefore energetically more efficient to live within your thermal neutral zone. You're not going to have to waste any energy on maintaining your body temperature. You can allocate it all towards mating, foraging, whatever. So given our hypothesis that heat is too energetically costly for northern flying squirrels, we predict that the northern flying squirrels start to energetically cool themselves at a lower ambient temperature than their southern counterparts, and are therefore forced to live outside their thermal neutral zone more often as temps rise in Maine. So we plan to continue our collection of metabolic rate data. This is not actually mine, but you can see that there's been a large cold weather bias, so we're coming at it from the other side. But we'll continue our collection of metabolic rate data under different ambient temperatures to determine kind of the break point between that thermal neutral zone and the energetic cooling for each species. Metabolic rate, however, is only part of the picture. A mammal can use energy to maintain its desired body temperature, or can save on the energy and just allow its body temperature to fluctuate a little bit. For this reason, well, this comes off a little crazy. For this reason, we are also concerned with measuring core body temperature. Last field season, we successfully deployed and recovered one body temperature logger. Hopefully this season, we are preparing to deploy and get back many more. But this slide shows the entire body temperature trace in the black line for the recovered logger, which was implanted in a male southern flying squirrel between August and November of 2017. The gray line indicates ambient temperature over the same time period. So you can see there is a cyclical fluctuation in body temperature associated with day-night, or the active resting phase, that stays consistent despite the pretty dramatic changes that we see in ambient temperature. If we zoom in a bit, we can see more clearly that cyclic pattern. The gray bars here then indicate nighttime hours, which is actually the active phase for a flying squirrel, and the white bars daytime hours, or their resting phase. Again, you can see that body temperature follows a steady cycle even when we have a pretty big variation of ambient temperature. From about September 27th to October 20th, we had about a 35 degrees Celsius change, and we really see that cyclical pattern staying consistent. We can also partition the values that we get for body temperature into daytime and nighttime readings to get an indication of mean values. So in this graph, the dark blue represents daytime, or again, resting phase body temperature, and the light blue shows nighttime or active phase body temperature. The squirrel has a higher body temperature during the nighttime active phase, as would be expected, but we also see that there is a lot more variation in this time period. All of this data, notably, does come from just one individual, so we're not making any conclusions yet, but we're excited to see how additional data will fit into this picture. So far we've talked about addressing coping with the heat by using energy to cool down, or by allowing your body temperature to fluctuate. One last option is seeking refuge from the heat. So flying squirrels nest in tree cavities, which are often known for their beneficial thermal properties. Beneficial in the winter, however. The pictured graphs come from a paper exploring the thermal properties of tree cavities during a Canadian winter. The open circles show ambient temperature, and the closed circles show temperature within a tree nest. So during the coldest parts of the night, you can actually see that the nest temperature is about 4 degrees Celsius warmer than ambient surrounding temperature. So what exactly does this mean for the summertime? Are tree cavities staying cool, or are they actually getting way hotter than the ambient temperature? How will this be affected by the changing tree canopy throughout the summer? We do not know the answers to these questions yet. Once we've caught some of our flying squirrels this summer, we are planning to tag them and hoping to locate their nest sites and monitor their experienced microclimate to answer some of these questions. So for those of you that were paying close attention at the beginning, besides questioning whether climate change could be a direct cause of rain shifts, I also asked whether these shifts were occurring further northward. When I began this project, everyone I talked to that was familiar with the area insisted that we would find northern flying squirrels here in Orono and Old Town. When I actually started trapping, I became extremely suspicious of this assumption. I had what appeared to be southern flying squirrels here, not northerns. So I thought, had Orono already made the transition between species that we saw happen in Holt Forest just about 2 decades ago? I can tell you that I was pretty certain that that had been the case. Then about a month ago, an undergraduate in my lab tested some of our DNA samples. She found some genetic markers from northerns and some genetic markers from southerns. To top it off, another undergrad about last week recently found a parasite known to innocuously infect southern species, but be lethal to the northern species. So needless to say, we have a few more things to figure out about the squirrels here in Orono. So stay tuned for more research coming out of the Lebec lab, hopefully to answer some of these questions in the future. And with that, I'll take any questions.
I spent a lot of time in the woods. How do you track a flying squirrel?
Trap or track?
How do you find them and follow them?
Well, first we have to trap them. So we actually have small mammal traps that we place on the ground. Even though they do travel from kind of treetops to treetops through their gliding, they actually forage on the ground for their food. So we bait with peanut butter and oats, which is a very tasty treat for a squirrel. And then we trap them in small mammal traps. Once we do have them, and we decide that they're like kind of big enough and adults, they're not necessarily a pregnant or lactating female, we can attach radio collars to them, and that is how we track them. So I'm out there in the middle of the day, which is when they're resting, so ideally they should not be moving, out there with a big antenna trying to follow the beeps and figure out where exactly this squirrel is. Granted, they are usually nesting somewhere in a tree, which is much higher above me, so I just try and triangulate to my best. Think, okay, it's probably in this tree, and then hope that I'm right. Hopefully, I'm going to dip into some of my friends in forestry that actually have the equipment to climb up to some of these nest sites, and so we can actually put some temperature loggers inside those nest sites and get at some of our questions.
Do they hybridize?
Yes, so which is also a possibility that we have thought about. So some hybrids have been found in southern Ontario and northern Pennsylvania. So it is I gotta claim up. Okay, so it is a possibility that that could be happening. We have to investigate the DNA a little bit more to determine that.
Next, we have Mark Royer presenting a tiny Java library for maintaining model provenance.
Hi, I'm a PhD candidate in computer science here at the University of Maine. My advisor is Chaw, and I'm going to talk about a library that we've developed that came out of work with Andre and Paul here at the university. Here's the outline of the talk. I'll give you a brief introduction of why we developed the library. I'll explain how it works on the Java virtual machine. I'll show you a small example, and then I'll give you some final thoughts. So the little Java library that we developed maintains the provenance of a software object model. And so when I'm saying model here, I'm talking about the way that objects are modeled in an application, a software application. And the way that this library works is that you can add the maintenance of the object provenance with only slight modifications to an existing Java program. And how this works is we take advantage of the Java annotation mechanism, and we use an undoable annotation. Then what we do is we extend the default Java class loader with our own class loader that can handle those annotations. The library that we've developed is very small. It's only about 50 kilobytes. And of course, the best thing about it is that you can apply it to existing Java programs with very little change. So why were we motivated to have this library, to create this library? When you're developing software applications in business, but also for scientific applications, oftentimes the requirements change. And when those requirements change, the application model has to change as well. Specifically in scientific applications, the software requirements change very frequently. And when this happens, the application developer has to go back and modify the model itself, but they also have to modify the program anywhere where that model touches. And it takes a long time, it's very tedious, and it's very error prone. So if we can have this happen in an automated way, then we can avoid some of that. So that's what this library does. This diagram is a diagram of the Java virtual machine. And the way that Java programs work is you write a Java program, it's compiled into Java bytecode. A Java virtual machine is a program that's run on a computer. And what that does is it takes your compiled bytecode and it loads it to execute the program. And that's what's shown up here at the top. After it loads your program, it's divided into separate areas. So the method area is the description of the program. The heap is where the dynamic objects are created while your program's running. And there are other areas for how the program gets executed. And finally, down at the bottom, there are also additional locations in the Java virtual machine that allow you to interface with native libraries on a system. What we do is we take advantage of this class loader mechanism. So we extend the loader, and when your program is loaded with those annotations, we recognize that in the bytecode stream, and then we inject additional bytecode that maintains the object provenance. So here's a diagram that shows how that loader works. We call it the undoable loader. Basically, you can go back and forth through the states, undo and redo. So we extend the default class loader. We override, we redefine the load class method, and we redefine the find class method. When we initially load the class, we determine if it's a user-defined class or not. If it's a user-defined class, then we look for those annotations. We inject the bytecode. Otherwise, if it's not a user-defined class, we just pass it on to the default class loader, and it's linked and loaded and run as a typical Java application. In order to maintain the state of the objects that are annotated, the model that you want to keep track of, we introduced two objects, memory manager objects. One's an in-memory manager, and the second one is a disk manager. And the way that it works is main memory is very fast compared to disk memory. Initially, when we keep track of previous object states, we keep it in main memory so you can go back and forth quickly. As that amount of memory becomes large, those states get pushed to the disk. And the amount of states that you keep track of and the amount of memory that these managers keep track of, you can set. So that way, if your system has less main memory or more disk space, you can set those parameters. And the important thing in this diagram is, so here's the main memory manager. More recent states are closer to the main memory manager here, and older states are pushed down to the disk manager with the oldest states furthest away. So I'm gonna give you a relatively simple example of how this works. So suppose that you have an application that does image processing. So maybe it's just, there's a function here that takes an image and it applies a filter on it. And we'd like to load multiple images, apply a filter, and be able to go back and forth based on what those images are. Well, to keep track of the image data, you might have a width, a height, and then you'd have the byte array that is the actual data for that image. And then you might have some additional transient data, which is just how do you display the image that comes and goes. So for our library, one of the changes to be able to keep track of the provenance is that the image model here is required to extend model interface. And what that does is the model interface implements a Java serializable interface, which allows us to use a mechanism in Java to traverse object graphs and convert them into byte arrays automatically. So then, so that was the model. And then you have the functions that you want to keep track of. So here's the most important thing about this slide. You have some filtering function, and it's already been written by somebody previously. And this is a pretty simple function, but it's still very difficult to tell what's going on just by a quick glance. But I'd still like to be able to maintain the image state before and after the function execution. The point of this library is that you don't have to modify this function at all. The only thing you have to do is append this undoable annotation up at the top. The rest of the function remains the same. The code to maintain the object state is injected automatically. Okay. So we have a small Java library that helps you maintain model provenance. It happens automatically as long as you annotate that a function is undoable. We use history managers to preserve the state. We use an extended class loader to inject the necessary byte code. And the amount of changes that you have to make to existing code is very small. You just have to annotate them as undoable, and you have to make your model extend that model interface. Now I'd like to just take a second and say, this technique is for Java, and it's a Java library, but it can also be applied to other virtual machine languages, such as C#. And you can use this as this technique in similar ways for scripting languages. I know that a lot of you are using Python, and Heather Clifford and I actually did some extensions in a similar way to this with some of the work that she was doing this last fall. That's all I have for you. I'd like to thank my brother, Dale Royer, for his input on this. And if you have any additional questions, I can bring up some examples on my laptop maybe after this talk. But thank you. Questions? Thank you.
Next, we have Keith Pontbriand with Excavations at Tranquility Farm, a seasonality analysis using mollusk remains.
Thank you, Will. Hope everybody's enjoying the symposium today. I know I've definitely been enjoying it. Today I'll be talking about my master's thesis research, looking at seasonal occupation at Tranquility Farm. And this research has been done in collaboration with Emily Blackwood. So if you're listening to this and thinking, this sounds kind of familiar, sounds like something we might have heard yesterday. This is in collaboration, so there will be some overlap. So just some bit of background and an explanation of importance. So why am I interested in looking at site seasonality? Understanding when people lived at these archaeological sites on the landscape gives us a better background for understanding their orientation to natural features and also their relationship to this Maine landscape. And when I'm talking about Native people, I'm talking about the Wabanaki people, the ancestors of today's Wabanaki in Maine. And I'm also really interested in learning about this because shell middens, as we've heard before, are a threatened resource. They're eroding, they are not going to be around for very much longer, so it's really important that we understand what information we can glean about them before they're no longer there for us to look at. A little bit of background, so thinking about some historical references. Originally, historical references indicated that Native Americans lived on the coast of Maine during the summertime and then moved inland during the winter. But in his 1996 paper, David Sanger proposed a two-population model. So there's actually one population that lives on the coast all year round, and then another population that lives inland all year round. And this is supported by looking at some of the very same types of clams that Emily and I are looking at for our thesis research, and looking at the growth rings in the soft-shelled clams, and also understanding that it is possible for them to live on the coast all year round because there are resources for them to exploit all year round. So the question that we're looking at through our research is, can we use oxygen isotopes to establish a season of occupation at Tranquility Farm? And the stable isotopes we're using are from the Maya arenaria clamshells. The site I'm looking at is Tranquility Farm, indicated by the red arrow there. Tranquility is located in Gouldsboro, Maine, which is conveniently located about a mile from my house. A little bit of background on Tranquility Farm. It was first excavated by Warren K. Moorhead in the 1930s. The black and white photo you see up there is an example of trench excavations, so they're not using the beautiful 1 meter by 1 meter test pits that we use nowadays. There was a brief hiatus in the excavations at Tranquility, and it was resumed in the 1990s and then again in the 2000s by the Abbey Museum in Bar Harbor. The picture that we have on the lower area there shows some tent posts. Let's see if I can get that going. So these are actually, these dark spots are post holes. Could have been from a wigwam or a fish drying rack. They're not entirely sure about that yet. So there's a lot of work to be done at this site, which is part of the reason I'm really interested in looking at it. We have one carbon date from the site indicating that it was occupied during the middle woodland period, which is goes back to about 2,000 years up until about contact period. And I did some research in 2016 for my undergraduate thesis looking at the faunal remains recovered from this shell midden. And I was looking to see if there's any types of animals present at the site that are only present during a particular season, and I didn't really find anything based off of that. So the proposed season for this site is either all year round or inconclusive. And that's when the oxygen isotopes come in. So to assess the oxygen isotopes of the site, we created two collections. The first of these, here, Emily's hands right here, are a modern collection of clamshells. This is something that we actually haven't seen represented in the literature before in this type of project, creating a modern collection of clamshells and then comparing archaeological samples to those modern samples. So what we did is collected clamshells from the historic clam flat middens in close association with the archaeological sites and created this modern record. The samples that I'm going to be talking about today, there's about 27 samples from the modern collection. For the archaeological collection, archaeological excavations had saved some of the clamshell remains, and I selected clamshells according to their location within the site, particularly trying to choose clams that come from areas that have other datable or other interesting artifacts. So for an example, taking clamshells that come from a soil sample or something that else can be carbon dated. So we have another part of the site that we're trying to look at with these clams. And for this, I looked at 13 clams. So with these clams, we went out to Iowa State University. Getting through airport security was interesting with all these clams. And used a micromill lab and mass spectrometer to analyze these remains. We're particularly interested in this last growth sequence on the clam. Analyzing that last growth will tell us what the oxygen isotopes in that particular section of the clam are, which will tell us approximately what the water temperature was during its time of collection or during its time of death. So we used two different sections on the clam. We used the outer edge of the clam, and then we used a more condensed version of this seasonal record, which is the chondrophore right here, which is a little tab on the inside of the clam. Both of those will show us what the oxygen isotopes were during its time of collection. So what did we find during this? In this lower graph right here, you'll see here we have the oxygen isotope value of the 018 value, and here we have our modern monthly collection. And each of these points indicates the average oxygen isotopes for each month of modern collection, with the little arrow bars being our analytical uncertainty. And you'll notice first that we actually have 3 months that we weren't able to collect the modern samples. Those were due to ice over in the winter and then red tide in the fall. So we have 3 times we weren't able to collect. Keep that in mind as we're going through this. And using these ranges of analytical uncertainty for each of the monthly averages, we then looked at those ranges and were able to place our archaeological samples within those ranges. So that's what you see over here. The blue is indicating the colder months and the green the warmer. And by placing my archaeological samples from Tranquility Farm within these ranges, we see that there is a concentration in the fall, the winter, and some in the spring, but not a lot in the summer right now. So this is indicating that Tranquility Farm is falling into this kind of 2 population model that Sanger talked about, where people are living on the coast of Maine all year round. And particularly, they're actually using this site at several different seasons. It's not just a 1-season site. So moving forward with these data, I'm particularly interested in adding to our monthly samples. For each of these monthly averages, we're using only 3 shells at this point in time, but we're gonna up that to about 6 shells so that we have a more robust monthly average. And we're also interested in cross-sectioning an entire shell, so looking at the oxygen isotopes from the beginning of its growth sequence all the way until when it was harvested. That will give us an idea of what the life of a clam looks like in isotopes. And finally, there's obviously the whole writing up of the thesis and all those things, so that'll be the easy part. And I'd like to give some acknowledgements to my collaborator, Emily, and also Dr. Alan Wanamaker, followed by Dan Sandweiss, Bonnie Newsom, and my other advisors, along with our funders, Abbey Museum, MAPI, and UMaine GSG. So thank you, and I'll like to take any questions. All right, thank you.
Next, we have Mariah Radu presenting the last termination recorded in Tsagaan Gol-Potanin, Glacier Valley, Altai Mountains, Mongolia.
Great, thank you, Will. And I'd like to thank CCI for providing my assistantship, which enabled me to work with Dr. Aaron Putnam and to do this research. This presentation is going to be a brief summary of the work that I did as a part of my master's, which I defended a couple weeks ago. Brief snapshot of what I'll be talking about. I'm researching the termination of the last ice age, and I hypothesized that glaciers in the Mongolian Altai, that they should retreat in response to the various mechanisms that have been proposed to drive deglaciation. Using beryllium-10 surface exposure dating and snowline modeling, I found that deglaciation, it occurred before the rise in atmospheric CO2 and during Heinrich's Stadial 1. Warming, it may have been the result of local insolation or the far-field effects from the tropics. I'm looking at a glacial valley in the Mongolian Altai, a mountain range which is located near the center of the Eurasian continent. It extends from Russia into Mongolia, and the climate is dominated by the westerly winds. Perhaps you've seen work by Peter Strand. He's developing a chronology around these lakes, and I'm looking at a glacial valley to the north. At the head of the Tsagaan Gol-Potanin Glacier Valley is the Potanin Glacier, the largest glacier in Mongolia. The meltwaters from this glacier, they form the Tsagaan Gol, or White River, which flow through the center of the valley. My research, we focused on 3 different sites in the valley: a down valley site next to Bayan Ul, or Bayan Mountain, mid-valley ice-molded bedrock hill, and the landscape just outboard of the modern Potanin Glacier. There's been little geomorphic work in the area, but the glacier was visited by a Russian geographer in the early 1900s, and he documented the glacier much closer to the little ice age moraines, with the ice terminating here, as opposed to in the modern terminating back there. In order to track the retreat of the glacier, I used beryllium-10 surface exposure dating, as well as geomorphic mapping to track where the glacier was in the past and what it was doing. And then I used snowline reconstructions to translate the extent of the glacier into a climate record. This is a key part of our research, is identifying glacial landforms, which are often laterally continuous, but very subtle vertically. Therefore, it's important to have high resolution elevation models in order to identify them. Think of what the LIDAR data set has done for identifying glacial landforms in New England. But this is the highest resolution elevation model for that down valley area, 30 meters per pixel. Fortunately, we brought a drone to Mongolia and used it with structure for motion to create digital surface models that look like this. So this is the same region. This is what was available, and this is what came from the drone. The level of detail is amazing, so it made it much easier to track the history of the glacier in the past through these subtle landforms. Next, I'm going to walk you through the surface exposure chronology along with the geomorphic maps to show you how the glacier retreated during the termination. The first site is at this Bayan moraine complex. And this is what it looks like from the ground. This photograph, it was taken from an outboard outwash plane looking at the furthest extent of the glacier when it formed a moraine, a terminal moraine. And on this glacial geomorphic map, to orient you, the picture was taken from the outboard outwash plane and looking at this outermost moraine. For this talk, I want you to focus on the moraine mapped in pink, because this moraine, it represents the local last glacial maximum. I've plotted the ages on a relative probability diagram, excluding the one outlier, and you see that these ages, they cluster into 2 populations at 23 and 28,000 years ago. So we interpret this moraine, Bayan 2, as a composite moraine that was formed during 2 periods during the local last glacial maximum. There are 3 glacial erratics that are inboard of the Bayan 2 moraine. They date from 22 to 19,000 years ago, and they bracket smaller discontinuous moraines. Inboard, the moraine was unstructured, which we interpret to represent retreating ice. The next area is Holy Mountain, this bedrock hill in the center of the valley. It's actually a UNESCO World Heritage Site where there are these beautiful petroglyphs, which I'm showing throughout the talk. That Holy Mountain, it records a history of thinning of the ice. To orient you, the vantage is looking to the east, and ice flow would have been in this direction. I plotted the ages with the age versus the elevation of the boulder. And what they show during the termination records 250 meters of ice lowering from 18.2 to 15.7 thousand years ago. So the third site is next to the modern Potanin Glacier. And this is what it looks like from the ground. Here's the Potanin Glacier. There's moraine in light gray formed during the Little Ice Age. And I want you to notice this obvious age contrast between the unvegetated moraine and this outboard landscape. To orient you on the glacial geomorphic map, this is the modern ice. With purple, I've mapped those gray moraines, and the orange dots, they represent glacial erratics that we sampled for surface exposure dating. In the red are the samples that pertain to the termination, and I plotted these ages on the relative probability diagram. And excluding the 19,000-year-old age, the other 7 ages, they cluster into 2 populations, at 17.7 thousand years ago and 16.2 thousand years ago. And here's another perspective on these boulders. So this is the lighter moraine. The vantage is to the east, flow is in this direction. And what these boulders they represent is a former ice margin. You can see on this glacially eroded bedrock that there are these benches. So this line of boulders, when they were deposited at by at least 17.7 thousand years ago, they represent an ice margin. But we can say more about these boulders. They have beautiful polish on them. They have striations. Therefore, we know that these boulders, they traveled through the glacier and they must have been deposited in an ablation zone where ice flow was divergent. So last step was reconstructing snowline in the past. Snowline, or the equilibrium line altitude on a glacier, it's where it's the line that divides the ablation zone from the accumulation zone, or at the end of the melt season, where is the snow deposited during that year. It's relatively easy to determine snowline presently, but how do we determine it in the past? And we use a method called the accumulation area ratio method. And this assumes that for a stable glacier, 40% of the surface area is in the ablation zone and 60% is in the accumulation zone. And we need to know the past surface of the ice, and I do this using an ArcGIS toolbox. Here's the snowline reconstruction for the last glacial maximum, and I found that snowline was at 2,680 meters. Did this for the Little Ice Age and found that it was 3,470 meters. But what we want to know is how snowline, how did it change in the past? And I used the assumption that the equilibrium line altitude is the same thing as the 0-degree isotherm in the atmosphere, which I both call snowline. I've shown you how I've developed it using the glacial reconstructions. I estimate it in the modern using a weather station that's next to the Potanin Glacier. And then I return to these boulders just outside the Potanin Glacier. As I said, they must be deposited in an ablation zone, therefore they are below snowline. I can use the elevation of those boulders as a minimum estimate for what snowline was in the past. I'm going to take you through cartoons that I made to reconstruct, to show you how we reconstructed the glacial history. At the local last glacial maximum, the glacier was at the Bayan 2 moraine, and this was snowline. But by 17.7 thousand years ago, the snowline had risen about 600 meters, as shown on the graph here. 15.7, we don't know what snowline was, but we know from Holy Mountain that the ice was retreating and that the climate was warming. We don't know what the glacier was doing during the Holocene, but that there was a re-advance at the Little Ice Age, and now this is modern climate. So as I said in the beginning, I want to compare the glacial history to my glacial record. And as you can see, that the deglaciation, it coincides with the rise in CO2. With the rise in insolation, excuse me. But that the warming, it precedes a rise in CO2. And in addition, that the deglaciation, it occurred during Heinrich Stadio 1. It's a period which Ali Balter discussed yesterday of cooling in the North Atlantic, but we see warming in the Altai, supporting the seasonality hypothesis. So what caused this warming? Possibly it was local insulation or far field effects from the tropics, which were also warming in this period. Here's a brief summary of what I have shown you today. And I just want to talk about the remaining questions of the research. What was the role of local insulation? Could it have caused all the warming during this period? Or do we need to call on far field effects, such as warming in the tropics, to explain it? And with my acknowledgements, I'll take any questions. Great. Thank you.
Next, we have Joe Pelto presenting Holocene deglaciation in Amundsen and Leave glaciers in Antarctica.
Thank you, Will. So today I'll be talking about my master's research. I work at 2 outlet glaciers in the southern Transantarctic Mountains at ice-free regions or nunataks like Mount Henson in the foreground or the Tusk in the background. The Antarctic ice sheet contains about 58 meters of global sea level equivalent. This is why it's crucial to investigate the ice sheet's behavior to help predict future sea level rise. And to do so, I'm investigating how the ice sheet responded, or the marine portions of the ice sheet responded to the major warming that occurred at the end of the last ice age. I work in the Ross Sea region, and today the Ross Sea is only partly covered by the floating Ross ice shelf. But during the last glacial maximum, the east and west Antarctic ice sheet expanded to fill the entire embayment with ice grounded to the seafloor. And as you can see in this reconstruction by Denton and Hughes. And so I'm focused on when the grounding line of the ice sheet retreated back to its current position in the embayment, which my results indicate occurred shortly after 3,000 years ago. So existing studies indicate the majority of retreat in the embayment occurred during the Holocene, with initial thinning by about 13,000 years ago, and more rapid retreat between about 9,000 and 7,000 years ago, with the grounding line position back to about Beardmore Glacier by at least 7,000 years ago. But there's still a lot we don't know about the nature of the recession in the embayment. Was the entire deglaciation one rapid event, or was the later portion gradual? And was it continuous, or did readvances occur? And so I work within a 400-kilometer gap in retreat data in the southern Transantarctic Mountains. So I hope to address many of those questions. And they're important to answer because understanding the pattern and rate of retreat will provide context for what caused the Holocene deglaciation. And further will help us to understand the recession history of the West Antarctic ice sheet, providing context for our future predictions of retreat. So I work at Amundsen and Leaf glaciers, which are both outlets of the East Antarctic ice sheet, draining its ice through the Transantarctic Mountains to the Ross ice shelf. I worked at Amundsen Glacier in December 2016 through January 2017 at Robinson Bluff and Watalis Peak, and at Leaf Glacier this past December through January at the 5 field sites I noted. And today I'll just be talking about the Duncan Mountains and Watalis Peak. So when the entire Ross Sea was filled with grounded ice, outlet glaciers such as Leaf and Amundsen were thicker than present. And once grounding line retreat begins, the glaciers also simultaneously begin to thin. As soon as the grounding line of the ice sheet retreats past the termini of the glaciers, that thinning has essentially stopped, and the glaciers are now close to their present elevation. So by working at ice-free regions or nunataks along the coast, we can observe that final thinning history of the glaciers when the ice sheet retreated past their mouths. The objectives of my work are to determine the former ice extent, elevation, and flow directions at Amundsen and Leave, and to develop chronologies for ice fluctuations and retreat during the late Holocene. And to do this, I first conduct mapping and sampling of field sites, and this allows me to create geomorphic maps of glacial deposits, which provide context for chronologies of ice recession. This was our first field site at Leaf Glacier this season, the Duncan Mountains. We worked in the Central Valley, which was surrounded by this high ridgeline, and at the bottom of the valley was this pond dammed by ice from the local glacier and from the ice shelf. And the next image is looking up valley. The valley that we worked in was covered in glacial drift, likely from the last two glaciations. And this is the location of the drift limit, likely from the last glacial maximum. And the positions below that represent when the ice was thinning back down to its present elevation. So I can use these sorts of deposits and observations to create my geomorphic maps. This is the location of that drift limit in the previous slide, and above that, mapped in orange, is the older drift deposit, which underlies the younger deposit mapped in red throughout the entire valley. And the pink represents the modern drift forming on the ice today. So these would have been deposited when ice was thicker than present and covered most of the Duncan Mountains. And as it receded, it would have left these deposits on the landscape. So these maps provide really important context for my chronologies of ice recession, and I'll be sharing my carbon-14 chronology today. So at my sites, glaciers dam ponds at the ice rock margin, and these ponds often contain seasonally open areas of water or moats where algae can live. So on the right are examples of different types of modern algae that we saw at the ponds in my field sites. And when Amundsen and Leaf glaciers were thicker than present, like the schematic of the maxima, they also dammed ponds further upslope that could contain algae. As the ice begins to retreat, there are two primary scenarios for the evolution of these ponds that reflect the timing and nature of ice thinning. And the first is a continuous retreat. As the ice thins, the ponds follow the ice margin downslope, and algae from the past ponds remain stranded on the hillside, recording their former position. So the algae represents a minimum elevation of that pond, as well as a minimum elevation of the ice necessary to dam that pond on a slope. So I would expect that dating algae from this scenario would result in a pattern of decreasing age with decreasing elevation, and that this continuous thinning of an outlet glacier indicates that the grounding line of the ice sheet is still to the north of its terminus. And the second scenario for retreat varies after the retreat stage. So once the grounding line retreats past the terminus of the glacier, it's essentially at its present elevation and is just slowly thinning back to the ice shelf. This can lead to an ice dam pond that persists in one location over a longer period of time. Given the scenario, I would expect that dating this algae would result in a spread of ages that represents the span of time the pond was in that position, with no clear age versus elevation pattern. And the slow thinning would represent that the glacier is close to its present stable position, and thus that the grounding line of the ice sheet has retreated past its terminus. So I dated 17 algae samples at Watalis Peak, and the bulk of ages are between 1300 and 2800 years before present. There's one younger age of 540 located immediately adjacent to the modern moat, so that young age makes sense. And I'm still considering whether the one old age of 5500 is real or an outlier. However, I'm suspicious of it because we ran the sample and got a different result, so I'm excluding that age in my interpretations for now. But overall, the range in ages that cover 1500 years support that second scenario of a slow final ice thinning of Amundsen Glacier. which would indicate the grounding line retreated past its terminus by the time of the oldest age, so shortly after 3,000 years ago. And I dated 15 algae samples at the Duncan Mountains, and they range from about 2,000 to 4,000 years ago. Again, that range supports the second scenario of slow retreat, where the grounding line retreated past its terminus by the time of Leaf Glacier, by the time of the oldest age, so by about 4,000 years ago. And this cartoon depicts the emerging story of ice retreat, grounding line retreat at my field sites. My results indicate that thinning at Leaf Glacier had essentially stopped and was close to its elevation by about 4,000 years ago, indicating that the grounding line passed by its terminus by that time, and that at Amundsen Glacier was close to its present elevation by about 3,000 years ago, indicating the grounding line had retreated past it by that time. So to conclude, most of the retreat occurred during the Holocene, with an initial period of rapid retreat between about 9,000 and 7,000 years ago. And the grounding line likely retreated past Leaf Glacier by about 4,000 years ago and past Amundsen Glacier by about 3,000 years ago. And the nature of the grounding line retreat appears to change sometime after 7,000 years ago. It appears to slow, suggesting the ice sheet may have begun to stabilize. And up next, I'm currently comparing my results with existing glacial histories to address questions such as what are the external forcing mechanisms for that initial rapid retreat, and why did the nature of the retreat change sometime after 7,000 years ago? And it's important to answer these questions as understanding the behavior of a past marine-based ice sheet, such as that which existed in the Ross Sea, will provide predictions on the response of the West Antarctic ice sheet to current sea level rise and increasing global temperatures. Thank you. Yes.
Is there any possibility of, I don't know the lithology of this area at all, but is there any possibility of dissolved carbonates in your pond that would have been in your ponds that could be biasing your to be more old than they are?
I don't think there's any possibility of carbonates, but to test this, we do take water samples at each site and precipitate barium hydroxide to find out how fast the melt is equilibrating with the atmosphere. And we do get modern levels for those states. Thank you.
Finally, we have Kate Warner presenting how do changes in the timing of ice out affect Arctic versus boreal lakes.
All right, thank you very much. So I'm going to talk about some work that was a collaborative effort between several members of our lab group, evaluating differences in the timing of ice out in Arctic versus boreal lakes, and thinking about the sensitivity of the lakes in the two different regions in terms of this ice out change. So changes in the timing of ice out are occurring throughout the northern hemisphere. So this goes back about 50 years, so this is a little bit older data, looking at freeze dates or the timing of ice on, and breakup dates or the timing of ice out. So as we expect, ice on is occurring later and ice out is occurring earlier. In the two regions specifically, this figure of the Arctic depicts the past 14 years, and they looked at about 13,000 lakes, and each dot represents a significant trend in the change and the timing of ice out. So all this yellow and red depicts lakes where we're seeing earlier ice out. And in the boreal region, these are a few lakes in Maine since about the 1850s roughly that have also experienced changes in the timing of ice out and that becoming earlier. And so as we would suspect, this change in ice out could have several implications for lakes in the different regions. And I'm only going to focus on a few of these today. So an important implication are the physical effects that we see. So in Arctic and boreal regions, if ice out occurs earlier, this could influence the length of spring turnover, influence the depth and stability of stratification, that warm mixed layer at the top, the length of the open water season, and how it's exposed to light. And then another thing I'll talk about today is how this might influence or have biological implications on the phytoplankton community. It also influences several other biogeochemical properties as well. And so another thing that we have to think about is, in addition to ice out occurring earlier, how this would be different for the different regions. So, and this is due to not only ice out itself, but also climate factors. And temperature would be indicative of changes or linked to ice out changes. So in the Arctic, ice out dates reflect April and May temperatures, whereas in boreal regions, these reflect February and March temperatures. And this could be important because ice out in Arctic lakes occurs between May and July, and therefore that would typically lead to a period of longer ice cover over the winter time, a shorter spring turnover season, and a shorter growing season relative to boreal lakes. And then the opposite, so in boreal lakes, ice out occurs between March and May, a bit earlier in the season, and when temperatures are cooler relative to the year, and then we would experience a shorter period of ice cover, longer spring turnover, and a longer growing season compared to Arctic lakes. And so our objective here was to look at the effects of ice out timing on thermal stratification and the biological implications during an early and a late ice out year. And so we have our two sites are here in Greenland, where we have a set of lakes. And then we compared this set of lakes to Jordan Pond and Acadia National Park. And so for the Arctic region, during the early ice out year, ice out occurred May 18th, which was 30 days earlier than our late ice out year, which occurred June 17th. And in the boreal region, ice out occurred March 19th for the early year and April 29th for the late year, so a difference of 41 days. And so there's variation in the lake mixing depths during these early and late years between Arctic and boreal regions. So when I say mixing depths, I'm referring to this warm mixed layer at the top or surface of the lake. And so here we see that during the both the early and late ice out years overall in the Arctic, we see shallower mixing depths, and in the boreal region, they're deeper. This may be due to patterns of solar insolation. So when the ice out occurs here in the Arctic in May and June, we're close to that peak solar insolation, so they occur relatively simultaneously. And then in the boreal lake, solar insolation is lower relative to the peak, and it has more time for that mixing in the water column, leading to deeper mixing depths. But we also want to understand why we might see variation during an early and a late ice out year, which could be due to a combination of factors, including that solar insolation and also including temperature. So in the Arctic lake during late ice out occurred 4 days prior to peak insolation when temperatures were warmer relative to the rest of the year, leading to really rapid stratification and formation of that warm mixed layer, contributing to shallower mixing depths. In the boreal region, the date of stratification was similar for both lakes. So during the early ice out year, we had a longer spring turnover period and warmer temperatures, leading to stronger stratification and shallower mixing depths. And so this could have several biological implications. What we focused on was looking at the phytoplankton community. And these species have strong associations with thermal structure, so they could be good indicators of this change. So this Lindavia botanica typically prefers deeper mixing depths. And this Discocels stylidra typically prefers shallower mixing depths. And we see that we have quite a difference in abundance of the two species. And in Arctic lakes, this Lindavia botanica is more abundant during the early ice out year, which is also similar to an ice out. It has that deeper mixing depth, so that makes sense. And in the Boreal lake, we also have more abundant during the late ice out year with, again, with the deeper mixing depths. The Steligera in the Arctic region, we have a more abundant during the late ice out year, which also has shallower mixing depth. So they seem to follow what we would expect in terms of their preference as indicators. In the Boreal lake, we found an opposite effect, and that could be due to a few factors. Jordan Pond is an extraordinarily transparent lake, so that may be influencing the abundance there. And so to wrap up, basically what we find is that the lake response varies, but it's really this timing of ice out in concert with the climate factors that influence the driving of the different lake mixing depths, which could have biological implications, and that there's important regional differences between the two areas, and that the length of spring turnover and duration of ice cover and growing seasons are also very important, and that, again, they have important biological implications, which would warrant further research. With that, I'll thank my lab mates and media for providing data, climate data.
So
Early.
all the break.
Thank you to my plan
Break
upper too.
till 10:15.